I just want to say welcome to all of you who are here joining us for the JDRF research update. We're really looking forward to this awesome panel discussion today. We have some really exciting and innovative people in the field who are joining us. So just before I introduce them to all of you, I want to quickly explain how today will be working. So this is a webinar, so we are going to be listening to some really great presentations, and there is going to be a portion at the end where you can ask questions and interact with our panelists. That's going to be at the end. There is a Q&A function available on Zoom at the bottom of your screen. You can press on that, type in your questions, and then we'll answer them at the end. At the beginning of this presentation, we will have some polls popping up with some kind of interactive questions for everybody. So that'll come up on your screen, you'll be able to answer, and it'll disappear when we're done. So today we are joined by four really awesome scientists that we're really excited to have here with us. So first we have Dr. Sarah Linkalater, who is the Chief Scientific Officer at JDRF Canada, and she will be moderating today's panel. So in her previous role, she helped to launch the Lancet Diabetes Endocrinology, now the leading clinical diabetes journal worldwide, and she became editor-in-chief in 2017. Sarah has over a decade of extensive experience as a professional scientific editor and carried out research on mechanisms of immune regulation at UBC in Vancouver and at the San Raffaele Telethon Institute for Gene Therapy in Milan, Italy. In her role as JDRF Canada CSO, Sarah will lead the organization's efforts to advance and expand its research partnerships and support its efforts to effectively engage the type 1 diabetes community. Also joining us today is Dr. Andrew Pepper, PhD, who is an Alberta Diabetes Institute scientist and assistant professor in the Department of Surgery at the University of Alberta, appointed to the Faculty of Medicine and Dentistry. He obtained his PhD at the University of Western Ontario while a member of the Clinical Islet Transplantation Program at London Health Sciences Centre, and a postdoctoral fellowship under the supervision of Dr. James Shapiro at the University of Alberta. Dr. Pepper is an early career investigator whose first academic appointment began in July 2018. His laboratory develops novel transplant modalities to improve pancreatic islet and stem cell-derived beta cell engraftment for the treatment of type 1 diabetes. Furthermore, his laboratory focuses on elucidating the relevant cell death pathways which contribute to beta cell graft dysfunction and T1D onset. He has over 10 years of experience in islet and stem cell transplantation in both the academic and industry sector, and has spearheaded several preclinical innovations that have gone on to phase 1 or 2 clinical testing. His work has received funding from numerous organizations, including JDRF, Stem Cell Network, Alberta Innovates, and New Frontiers and Research Fund, and the Canadian National Transplant Research Program. Recently, the international standing of his work was highlighted by being the recipient of the JDRF International Career Development Award. He's also an active member of the Transplantation Society, International Pancreas and Islet Transplantation Association, Canadian National Transplant Research Program, Cell Transplantation and Regenerative Medicine Society, and the JDRF Beta Cell Replacement Consortium. His project title is Clinical Islet Transplantation: Is the Future Finally Now? Also joining us today is Dr. Cathy McCoy, who completed her PhD in immunology at the Milan Institute of Medical Research and Otago University in Wellington, New Zealand. She then completed postdoctoral training and was a junior group leader at the Institute of Experimental Immunology, University of Zurich in Switzerland. In 2006, Cathy was recruited to McMaster University in Ontario as an assistant professor, where she held a Canada Research Chair in Gastrointestinal Immunology. Cathy then moved back to Switzerland in 2010 and joined the University of Bern, where her lab studied the role of the intestinal microbiome in regulation of the immune system. In 2016, Cathy was recruited back to Canada, where she is a professor in the Department of Physiology and Pharmacology in the Cummings School of Medicine, member of the Snyder Institute of Chronic Diseases, and director of the International Microbiome Center at the University of Calgary. Her research focuses on the dynamic interplay between the gut microbiota and the innate and adaptive immune systems, and aims to understand how exposure to intestinal microbes, particularly during early life, influences immune development, immune responsiveness, and susceptibility to diseases. Her project title is Can the Intestinal Microbiome in Early Life Impact Susceptibility to Type 1 Diabetes? And finally joining us, we have Dr. Bruce Perkins, MD, MPH, who is a professor, endocrinologist, and diabetes complication clinician-scientist at the University of Toronto, who is appointed to both the Faculty of Medicine and to the Institute of Health, Policy, Management, and Evaluation. He holds the Sam and Judy Pencer Family Chair in Diabetes Clinical Research. He obtained his MD in internal medicine training at the University of Toronto, then went to complete his endocrinology subspecialty training at Harvard University, his Master's of Public Health in Epidemiology at the Harvard School of Public Health, and a research fellowship in epidemiology at the Joslin Diabetes Center. Using longitudinal cohort methods as well as clinical trials, his research work is focused on early biomarkers of diabetes complications and interventions for the prevention of complications, including artificial pancreas technologies and disease-modifying adjunctive-to-insulin pharmacotherapies. In 2015, he was awarded the Canadian Diabetes Association CIHR Young Scientist Award for his research. Among other projects funded by the NIH, JDRF, and Diabetes Canada, he leads an Innovations in Type 1 Diabetes group within the Diabetes Action Canada, a national patient-oriented research strategy. Short research projects as topic title is, Can we maximize time and target by combining closed-loop insulin pumps with adjunctive drugs, the Class 17 study. So I'm now going to pass it over to Dr. Sarah Linkalater, who is going to take over this panel conversation. So Sarah, take it away.
Good morning, everybody. I'm just going to share my screen here. Give me a moment. Can everybody hear me okay? Okay. All right. How's that? Great. Okay, so thank you everybody for joining this webinar. It's really my pleasure to be presenting here at the Connected in Motion Slipstream event. As we've already heard, I work with JDRF Canada, and I'm delighted to be convening this panel of experts across the field of type 1 diabetes. And so we'll dive right in with our agenda. We're going to engage everybody tuning in with a quick icebreaker quiz. I'll then give an overview of the field of type 1 research to sort of set the stage. This is really only about 3 or 4 slides, because really then we have the main event coming from our panelists, Dr. Cathy McCoy, Dr. Andrew Pepper, and Dr. Bruce Perkins. So if I could ask our tech support to put up the quiz, then we'll get everybody answering some questions. So I actually don't know how this works at your end, but if you could have a go at answering the first question here, what causes type 1 diabetes? So is it A, genetics, B, a viral infection, C, stressful life events, or C, the causes are not fully understood? We'll give you about 5 seconds to answer those, and then I think we get results popping up on the screen. Oh, wow. Okay, we have a very well-informed audience here. I think this quiz is going to be super easy. Sure enough, we know that the causes of type 1 diabetes are not fully understood. Probably the causes are multifactorial and might differ from person to person. So next question. When was insulin discovered? Was it 1821? Was it 1876, 1921, or 1945? Okay, again, everybody's on par. It's 1921. We are approaching 100 years since the discovery of insulin, and we have lots of reasons to celebrate that because that happened right here in Canada, in Toronto. So it's a very important time in type 1 diabetes research. So on to the next question. Okay, what are the main cells in the body that make insulin? Alpha cells, beta cells, delta cells, or PP cells? I have a feeling we're going to do really well on this one. Okay. Yep, we are all very well aware that the cells that make insulin in the body, for the most part, are beta cells in the pancreas, but those are all different types of endocrine pancreatic cells. Okay, I think we got 1 or 2 more questions left. Here's a slightly trickier one. What happens in the early stages of type 1 diabetes? The immune system starts attacking beta cells, or beta cells start to misbehave and send out danger signals, or both of the above. So a couple seconds to answer those questions. Okay, so most of you were correct. Probably both of the above happened. This is something we've more recently begun to understand about type 1 diabetes, is that beta cells are not just a passive victim that get attacked by the immune system, but probably something that happens early on is they play a role in their own demise by sending out danger signals or behaving in a slightly different way that attracts the immune system. Okay. Do we have one more question, or are we all done? One more question. Last one, how many beta cells does someone with clinical type 1 diabetes have? Is it 0? They're all destroyed by the immune system. Lots, the body just can't respond to the insulin that they make anymore. Or it depends. I'll give you a few seconds for that one. Okay. Yep, correct again. It depends. The reasons why the number of beta cells that somebody with type 1 diabetes has really depends on many factors, including how long you have had type 1 diabetes for, but also several other factors. And you might be surprised to learn that many people that have had diabetes even for decades still have existing beta cells. Perhaps they don't make insulin anymore, but sometimes there are still some there. So that has shifted how people are approaching the search for cures to type 1 diabetes. So we'll close that and we'll dive into the presentation now that everybody's well awake. It seems that everybody's very well informed already on the topic of type 1 diabetes research, so I think this presentation is going to be very exciting when we come to the Q&A. So just to give you a quick history of some of the highlights in type 1 diabetes research, I think it's important to talk about where we've come from before we talk about where we're headed. So although we've known about diabetes for centuries, technically it is thought that it's really just classified as a clinical entity in 1812. As we've already discussed, insulin was discovered in Canada in 1921, but it was really not until the 1970s that we really began to characterize type 1 diabetes as an autoimmune disease, because this is when we really began to develop the molecular tools that allowed us to really study what was happening inside the body. And since then, there have been very rapid developments in different scientific or clinical advances that have allowed us to treat and study type 1 diabetes much more effectively. We've got HbA1c introduced into clinical practice in 1977. And more recently, we are making major strides forward in beta cell replacement. And we'll hear a little bit about that today from Dr. Andrew Pepper. So some other advances that have really shaped how the field is approaching better treatments and cures for type 1 diabetes. We've learned recently that type 1 diabetes is highly heterogeneous, and that means it's quite different in different people, and there may even be different subtypes of disease. So this is really directing how we search for cures. It's not going to be a matter of one size fits all. We've also learned that type 1 diabetes probably starts weeks, months, or even years before clinical diagnosis, with asymptomatic things happening inside the body. Beta cells play an active role in the disease. We've already talked about how they don't simply they seem to trigger the immune system as well as being attacked by it. We've also learned that beta cells can be grown from stem cells in a petri dish, and this was first shown in 2015 in human cells, and there's been major advances since then, and now we are in clinical trials with several of these products that may be a beta cell replacement opportunity. We've made major strides recently, of course, in technology. We now know that technology is remarkable for reducing the burden of disease management, and things like artificial pancreas systems are developing very quickly, and we can expect to see a lot more coming onto the market in the next few years that will make managing disease easier. And finally, although we've known this for a long time, the idea that addressing psychosocial issues for good diabetes management is gaining prominence rapidly and is being incorporated into every type of study that's being done in the clinic. So that's also an important advance. So this is sort of my last main slide to set the stage, and that's just to talk a little bit about the stages of type 1 diabetes. More recently, it has been discussed that diabetes sort of is classified into 3 stages. At the beginning of the process, a person may have a variable genetic and environmental predisposition for type 1 diabetes, and at some point there is an event that triggers an autoimmune response that may be genetic and may be environmental. It's probably a combination of both. And in stage 1 disease, a person is having active autoimmunity taking place and developing autoantibodies, which are biomarkers in the blood that can tell that a person is very likely to develop clinical type 1 diabetes within about 10 years. In stage 2, those autoantibodies persist, and there also begins to be the first signs of glucose dysregulation that could be detectable with a blood test, but probably not any clinical symptoms happening just yet. At the time of diagnosis, beta cells have begun to be lost through autoimmunity, and the demand for insulin by the body exceeds what the beta cells remaining can produce. And that's when we rely on exogenous insulin and diagnosis takes place. So what we talk about as clinical diabetes is what is referred to as stage 3, and using this sort of paradigm for understanding stages of diabetes is really affecting how we approach the search for cures. So in these colored bars, I've showed that in the area of prevention research, we are looking to intervene in stage 1, stage 2, or even a little earlier in disease at people that have genetic risk. If we look into cures, so beta cell protection, beta cell replacement, or immunotherapies, we're focusing on different stages of disease as well. So the search for cures is really covering all stages of diabetes. And a cure for someone with stage 3 disease may look different than someone who is at high risk, so in stage 1 or stage 2. So the research goals in the type 1 field are pretty clear. They are to prevent the disease, to stop type 1 diabetes before symptoms start in people that are high risk, to cure the disease, so to stop and reverse autoimmunity by targeting either beta cells or the immune system or both. And third, to come up with better therapies to make life with type 1 diabetes easier, healthier, and safer until cures become available. And this is through either therapeutics or technology. And so I'm grateful to have with us today a scientist who is working in each of these fields. So we're going to start with Dr. Cathy McCoy, who's going to speak more on the risk side of type 1 diabetes and her work in the microbiome. So I will stop sharing now so that Cathy can begin. So we're going to have each of our presenters sharing for about 7 minutes, and then we'll move on to a Q&A. So thanks, Cathy. Take it away.
Okay. Oh, thank you very much, Sarah. Can you all see my screen? Okay. So I wanted to talk to you about an introduction to the work that we are doing, trying to understand how the microbiome could play a role in the impact of the susceptibility to type 1 diabetes. And the reason that this is, we feel that this is important and interesting is because I think probably everybody here really understands that genetics does not explain all of diabetes. There are definitely environmental risk factors or environmental cues that are playing a role. We really just don't know what those environmental cues are or the importance of them. We know that there's environmental risk if migrants move from one country to another country. They assume the same risk for type 1 diabetes as the country they move to. And in Europe, there are some really good examples of the risk of type 1 diabetes differs in really, really close areas. So where the people are quite genetically the same. So Finland, Russian Karelia, and Estonia. So it's really, these are separated by socioeconomic borders, basically. And Russian Karelia has a very low incidence of type 1 diabetes, whereas Finland has really one of the highest in the world. The type 1 diabetes risk has become more homogeneous as there's been greater movement of people around the world. And only 10 to 15 people that have a genetic risk for type 1 diabetes actually develop it. And if you look at genetically identical twins, if one twin has diabetes, the risk for the other twin is less than 50%. And that really tells us that there is a dynamic interaction between the genes and in our environment. Now, if you say environment, that is a huge word. There are so many things that can bring about the environment. And you have to sort of think about the environment probably in the last sort of 60 years. What has changed in our environment in 60 years that has led to a really large increase in the incidence and lowered the time of onset of type 1 diabetes, but also other immune-mediated diseases. You can think about our diet has changed hugely. You can think about our lifestyle, our hygiene, how we live. You can think about the huge increase in antibiotic use. We like to think that actually all of these are probably important, but they also all lead together to have an impact on one environmental determinant that we feel is very important, and that is the microbiome. We think about the microbiome as kind of an interface between the inside of our body and the outside environment of that we live in. It's kind of like a rheostat that takes information from our environment and relays it into our body. Now, our microbiome is the microbiota that inhabit our body. And now our microbiota, our body is colonized by viruses, bacteria, fungi, and there's probably 50% of the cells in your body are actually microbial. So only 50% of the cells in your body are actually human. And if you think about genes, that there's way more microbial genes in your body, so genes that are expressing proteins and function, there's more than 10%, less than 10% of those genes are actually human eukaryotic genes. So we really think that this microbiota is very important. And that's because it has huge functions. One area that the microbiota is very, very highly represented and very many microbes is in your gut. And this intestinal microbiome, we know, has incredibly important functions, protects us from pathogens, it extracts energy from our food, it produces essential vitamins that we can't produce for ourselves, it helps to regulate our metabolism, and it educates and regulates the immune system. And we believe that this last one is incredibly important. And it's very important early life, but it's also important throughout life. Your microbiota probably sets the tone of the immune system and sets the threshold that the immune system is going to react. Now, early life is very important. So how does your microbiome develop? We believe that babies in utero are in a mostly primarily sterile environment, and then they get colonized with the external microbes when they enter the world. They enter a microbial world. So already starting in the birthing process, they start to be colonized with microbes. The most important place for these first microbes to come to seed this newborn baby is from the mom. So we cannot just look at an individual's microbiome. We have to think also about the mother's microbiome. And the most important thing is that the mother should be transmitting through vertical transmission the microbes that baby needs at the very beginning stages of life. Now, we and others have done a lot of work on understanding how early life microbiome shapes the development of the immune system. So we know that even during gestation, so when the baby is in the uterus, which we believe is sterile, this baby is still affected by the mom's microbes. And that could be her intestinal microbes, because even in utero, these microbes, they are, when the baby's in utero, the mom's microbes are metabolizing her food, and they're making small molecules. And these small molecules, these metabolites, are able to enter in across the placenta. And that's where they start the functional shaping of the newborn's immune system. And then these signals, they come to the baby in utero, but they also then, after the baby is born, you get vertical transmission from the mom's microbes, and you also get transmission through the breast milk. And the breast milk also helps to shape that early life microbiome. And then early life immune development and neurodevelopment happens throughout probably the first 3 years of life, where the immune system is still undergoing development. So my work, my lab has really been very interested in understanding how both the maternal microbiome and this really early life microbiome, how these two act together to shape the development of the immune system, and also how that impacts on the pancreas in very early life. We're wondering whether some of these signals coming from the microbiome, may be shaping I'm trying to stop my timer, how these two work together, and maybe they are also shaping the pancreas and shaping maybe some of those early signals that the pancreas may be giving out that then alert the immune system to try that started that early attack. So we're really looking at immune development, metabolism, and how that together impacts on type 1 diabetes. And I'll stop there. Thank you.
Okay, thanks, Cathy. That was fascinating. We're going to save up questions, so please, those of you that have questions for Dr. McCoy, please enter them into the Q&A, and we'll do them all at the end during the panel. So moving on, Dr. Andrew Pepper, we'd love to hear your presentation on beta cell replacement.
Thank you, Sarah. I'll just share my screen here. Hopefully you can see that.
Yep.
Well, thank you. It's an absolute pleasure to virtually be here to share with you all an update on clinical islet transplantation, as well as some advancements in the field, and hopefully stimulate a discussion. Is the future finally now? Are we at the forefront of what we can do? So to step back as a matter of perspective, what is islet transplantation or beta cell transplantation if these are derived from stem cells or animal sources? Well, in the context of clinical islet transplantation, it starts, like all transplants, with a precious donor. These are cadaveric human donors, and we procure the pancreas. We remove the pancreas. It sits just below the stomach and nestled into the small intestine. And we isolate the 2% of the pancreas that is responsible for the endocrine function, these islets of Langerhans. So essentially, we mash up the pancreas with enzymes and some mechanical digestion, and we free up this 2% of the mass, and we transplant it back into the recipient's liver, where they embolize, hopefully engraft, see glucose and secrete insulin, restoring physiologically relevant glucose control. And we look at the impact of the different therapeutic options have on glucose control. If we compare, one of the predominant forms is the artificial pancreas. And this is like a bionic pancreas where you have insulin pump and sometimes tethered to other hormones with a continuous glucose monitor. And we can see this patient's blood sugar profile before the artificial pancreas is set up into this patient. They have quite a bit of flux in their blood sugars going from high to low. And then after the artificial pancreas is in place, we can see an improvement in glycemic control, but by no means is it physiologically perfect. That would be in that gray zone. And this isn't all patients, but that some have this still glucose liability. If we look at a patient that is a candidate for an islet transplantation, these ones are generally very brittle type 1 diabetics. It's a last resort. They failed on continuous glucose monitors and exogenous insulin therapy as a whole. So in this patient's blood glucose profiles, they have quite a bit of periods of hyperglycemic, but more concerning are these periods of low hypoglycemic events. And these can be deadly if undetected, especially during the nighttime. So this person had a transplant, and 9 months post-transplant, we can see that they have totally restored their physiologically relevant glucose homeostasis. Now, again, this is not a perfect application and therapy, and is really limited to those that are hypoglycemic unaware and have failed on all other therapeutic interventions. And also this function is not always durable. Some patients require 1 or 2 or even 3 transplants, and there is attrition over time. So right now, about 50% of individuals that are transplanted are completely off insulin at 5 years. So there is a lot of room to improve. And when we look at the hurdles that face the islet graft and islet transplants in general, we can start right at the beginning. We were really not, before we even transplant, we're not really sure what islet prep is the most functional. We really lack a good potency or viability assay before we transplant to say, yes, this preparation has a good chance of restoring glucose control to the recipient. And also during the isolation procedure, through the enzymatic and mechanical digestion, we strip the islets from their natural neural and vascular networks. And as a consequence, when we put them into the liver, they become poorly vascularized and hypoxic. So we need to look at alternative transplant environments to put these cells in. And when we put them in the liver, it evokes a massive inflammatory response, generally platelet and complement driven, because these islets are now bathed in blood. And it's been estimated about 70% of the islets that we transplant are acutely rejected within hours to days. And as a result, we need at least 2 donors to treat 1 recipient. And like all transplants, there's immune reaction when it's the adaptive immune response. It's alloreactive. So the recipient's body is going to notice the tissue as being foreign and stimulate a response to reject that tissue. But type 1 diabetic patients are also an interesting transplant population because they have an occurring autoimmunity. Their body has obviously seen their cells as foreign and has destroyed those islets. So when we transplant, we also have this autoreactivity to the insulin-producing cells that we've transplanted. Again, as a way to combat this, the patients require lifelong chronic and systemic anti-rejection drugs to prevent the rejection of these beta cells. However, the drugs that we actually use in clinic are diabegenic, so they actually destroy the beta cells as well. So taken together, this really limits the patient inclusion to the risks of a lifelong immunosuppression have to outweigh the benefits of glucose control. As a result, the poor islet graft dies over time. So in the lab, we're trying to implement futuristic innovations today to keep these islet grafts happy as long as possible. So one approach, instead of using systemic chronic immunosuppression, postdoc in my lab, Prue Kapan, is looking at localized drug delivery of immunosuppression. And essentially what we do, we have these bubbles, these me cells that will elute drugs of interest, and we mix that up with the islet preparation and transplant it into the recipient. And we're hopeful that this will reduce and potentially even eliminate the need for systemic immunosuppression. The next, we're looking at alternative transplant sites. Instead of relying on the liver, we're looking at the area underneath the skin. And Katerina, a PhD student in my lab, is trying to recapitulate the natural pancreas environment, but underneath the skin in a non-invasive way. And we're starting a clinical trial, a pilot clinical trial in Edmonton, looking at this site as a potential surrogate to the liver. Next, we're also looking at novel cell death inhibitors. So as we understand why islets die during type 1 onset, as well as post-transplant, we can look at developing pharmacological inhibitors to target these non-traditional cell death pathways. And one area of active research is the regulated necrosis. And we can see that this had a big impact in ischemic reperfusion injury, and we think it will have an impact in islet transplantation. And finally, stem cells. Our lab, as well as many across the world, are looking at recapitulating the natural pancreas development, but in the petri dish, using stem cells, either derived from yourself or from an alternative person. And we can now not rely on precious human donors. And one trial going on in Edmonton is taking these cells and putting them in an immunoprotective device. So this device has a physical barrier. It doesn't allow immune cells in, but allows a passive diffusion of insulin out and glucose in. So we're very hopeful that we'll learn from these trials. And taken together, as we implement these innovations today, the lessons learned from these early clinical trials will hopefully transition islet and beta cell transplantation from a therapy for a select few to the mainstay treatment course and option for all type 1 diabetic patients. So I'll thank you, I'll stop there, and really thank the JDRF support as they're funding all these projects, and look forward to having a discussion afterwards. Thank you.
Okay, thanks, Andrew. That was fantastic. Really exciting. So again, please save your questions, and we'll move on to our final presentation from Bruce Perkins before we open it up to Q&A. Thanks, Bruce.
I've learned repeatedly that you need to unmute before sharing, and I just made the error again. Sorry about that. There we go. All right. Well, first of all, I've enjoyed this a lot so far. It's really great to hear from Cathy and Andrew. This is phenomenal research. I've learned a lot from those few minutes alone. Babies come from eggs, islets smile, like it's really motivating. Thank you so much. I love it. So, you know, we already heard that it was nearly 100 years ago that insulin was discovered by Banting and Best and their team. And this, you know, I'm eternally grateful as a person living with type 1 diabetes as well. I owe everything to them. But if they were here today with us, there's a few realities that I think a lot of you on this call know. That people today with type 1 diabetes, they're, you know, have the reality of many feeling isolated, dealing with a very high degree of self-management burden, fearing hypoglycemia, fearing complications in the long term. And the part that kind of really breaks my heart, to be honest, despite all these really promising therapies that we have, is that it's still actually a small proportion who meet the type of blood sugar control targets that we feel prevent the long-term complications. It's actually, like in the US, we know it to be 1 in 5 people with type 1 diabetes who would hit that A1C or time in range target that helps us to prevent long-term complications. And, you know, it's not really surprising if we take a critical look. Like, I know that a lot of you on this call may use sensors and it's changed your life, or a pump and it's changed your life, or a hybrid closed loop. But if we look specifically at what A1C or time in range benefit these things have, the evidence for an insulin pump therapy, if you randomly assign people to it, at the best, it's probably a 0.3% better A1C than being on injections. And actually more recent trials don't even suggest that. Or using a continuous glucose sensor, it's an amazing tool, but on average, the studies tell us that an A1C is about 0.3% better. And then hybrid closed loops, similar, A1C of 0.26% better, or a time in range of about 10%. So why not, if, for example, there are drugs that we use and that have been transformative in people with type 2 diabetes for blood sugar control and other metabolic control, and frankly, even transformative in terms of reducing even complications like kidney disease, cardiovascular disease, and even death, to be honest, in type 2 diabetes. If they work so frickin' well in people with type 2, why the hell not try them and quickly, like really efficiently study them in people with type 1 diabetes? And so an example would be the sodium glucose linked transporter inhibitors. These are medications that overcome a thing that insulin on its own doesn't do. Even someone with great blood sugar control will have some periods of time in their day or week or month where sugars are higher. It upregulates these transporters that makes the kidneys take back sugar that would otherwise be lost in the urine. And the reason that's important is that from an evolutionary standpoint, we wouldn't have survived if we could lose calories in our urine. So it's very, it gets upregulated to take in more sugar. So blocking those, at least in part, helps us to control blood sugars and lose some of that glucose in the urine as an add-on to insulin. And actually, these therapies are not approved in Canada or the US, but have been approved in Europe and in Japan. So, you know, in our hands, I thought, well, all I care about with people with type 1 is that if we have things that have a biological effect on lowering glucose, that people get the choices of these. You know, not everyone uses a pump. It's actually in Canada, it's shockingly small proportion, or sensors. But different people have access to them, and I hope everyone will have access to these eventually. But even those who have access don't all choose it. The point is, I want to have a panel of choices to help every individual have their best chance of getting closer to target A1C. So in this study that we've just completed and haven't published yet, what we did is we took people with type 1 diabetes and brought them in for a series of days where we had them eat meals in the clinic. And on some days they were in open loop therapy, so a pump and a sensor. Others were on a closed loop system. We collaborate with the McGill Artificial Pancreas Group. And other days they'd be on a placebo pill, and other days they'd be getting that sodium glucose linked transporter. And you can see that the artificial pancreas, it's in the red line, is the average sugar over a day. They do pretty damn well. They get 65% of that day in target. But when you do the same thing and add on this medication, empagliflozin, the sodium glucose linked transporter inhibitor, you can see they do even better still, 86%, which is great. It ain't like that example that Andrew gave of that one, you know, the few post-transplant patients who go back to normal glucose, but we're really getting there. So the class 17, it stands for Closed Loop Assessment Study 17. It's a study where we're using a type of pump, it happens to be called a tandem pump, and a type of sensor, happens to be a Dexcom sensor, and using an Android phone that carries in it an algorithm that's a hybrid closed loop. So it's automating insulin delivery all the time, but it depends on the patient, the person, to signal meals and be as good with the insulin for meals as possible. So our best closed loop systems now are hybrid closed loop. They still rely on people carb count or estimate their food bolus for meals very well. But now what we're doing is they're free living over the course of months in this design. It looks so complicated, and I probably shouldn't have even shown this slide, but the idea is that we want them to be on an artificial pancreas or in the conventional therapy open loop. And at the same time, we want them either to be on empagliflozin or on a placebo. And we want everyone to experience each of these scenarios. So now mathematically, you realize you have to randomize people down different pathways to be on one or other of the drug and one or other of the device. And they're actually on this over the course of about 5 months in total. And we're in the midst of this. We've now recruited about a quarter of the patients for this trial. There'll be close to 40. We're a bit slowed down by our COVID efforts, but we're still maintaining contact with these folks. And my hope is that this kind of research, just at a practical level, helps us to figure out what are the best combinations of therapies that people with diabetes could have access to give them the best chance of better blood sugar control. So maybe I'll stop there. I have no idea, Sarah, how many minutes I took.
That was perfect. Thank you, everybody, for sticking to time. That gives us almost 20 minutes for questions, so that's great. So I just wonder, can our panelists see questions or only me? I think it doesn't matter, and I'll just dive in. So we have a lot of questions. I'm hoping we'll be able to make it through most of them. So I'm going to prioritize those that were directly related to the discussions that we've had, and I hope that there might be a mechanism to answer any that we don't get to through Connected in Motion after this event. So I'll take one from an anonymous attendee that asked, when we talked about the 3 different stages of clinical type 1 diabetes, how long can one stage last? It probably varies a lot from person to person, and in fact, the natural history studies that we have, I don't think we exactly know. And a second part to that question was, if someone was diagnosed at 20 years old, would they be considered stage 1 from the time that they were born? Probably not. Autoimmunity starts probably in early childhood, but again, this really varies. Prevention research is trying to pin down the best time to screen children. So at what age are we most likely to pick up people in stage 1 at a time when we can begin to think about interventions? So the answer is, I think research is still in progress, but the diabetes disease pathway probably starts sometime in childhood, certainly for people that are diagnosed in later childhood. Okay, so we'll move on to ask some questions of Cathy McCoy. Cathy, first one is, if there might be any symptoms, say gastric symptoms, that a mom might have in pregnancy that may give an indication that their child could be predisposed to disorders including type 1 diabetes, or if you can look back at pregnancy and see any correlations. So it's a big question.
So I am not aware of any studies or any thing that has shown that you could have symptoms during pregnancy and then you think, yes, my child is predisposed. I don't know of any studies that have shown that. What we do hope to be able to do, and people are doing this now, is to try to do studies where we are sampling or collecting the microbiome from the mom and then from the baby early in life. So collecting throughout gestation from the mom and through the baby, and measuring what microbiome, what bacterial species or fungi or viral species are there. And then we have to wait over time to see what is there, how that associates with probably a genetically at-risk child, whether they develop type 1 diabetes and at what time point. Those are association studies. So we with those studies, we still cannot say what is causal. And that's one of the biggest challenges we have right now in this field, is that we can study people with type 1 diabetes and can study and look at what composition of the bacteria is, and many studies have shown it may be different, but that doesn't mean that microbiome changes were causal. And because microbiome changes with disease states as well, changes with diet. So we do really have to do these early life studies and look for these types of associations.
Okay, thanks. We'll come back to you, Cathy, because there are other questions, but we'll move to Dr. Pepper now and we're going to ask 2 short questions about islet transplantation. First who are the candidates for islet transplantation? Who could be ruled out? And actually, we'll go with that first.
Oh, thank you for the question. Sorry I couldn't get into detail on that. Every center will have a little bit different inclusion criteria, but generally in Canada, they'll have to have failed on self-administration of insulin as well as with a continuous glucose monitor. There's a wonderful paper in Diabetes Care by Peter Sr. that structures out the 4 stages and where an islet transplant recipient would fit into that. The indications or who might be ruled out are generally people that have pretty good control, so the risk of immunosuppression wouldn't be warranted in that case. And normally these patients are 20 to 30 years of poorly controlled T1Ds, and the main thing is hypoglycemic unawareness. Because this is a deadly complication, so the risks of lifelong immunosuppressions absolutely outweigh the benefits outweigh the risks because we can completely restore those hypo events even with partial functions.
So related question on the hypo unawareness. Do those who have received a transplant become hypo aware again at some point? This would, let's say they are still requiring to take some insulin, maybe less.
Right, as like scientists, and I'm sure clinicians feel the same way, the holy grail is to have long-term durable insulin independence. However, that is not always achievable, but there is some wonderful metabolic advantages of just having some form of function. And we measure that through C-peptide. So this is secreted in equal molar to the alpha and beta chain, the active forms of insulin. And we know the C-peptide has to come from a cell. So a patient post-transplant, they may be on a few units of insulin a day, but they have some C-peptide positive. And as long as you're C-peptide positive, you completely eliminate from what we've seen in long-term studies, the hypoglycemic unaware events. And so at least at 1 year, almost 100% of the patients are completely hypo-aware. And in a recent phase 3 clinical trial conducted in Canada, the United States, 80% of the patients at 2 years met this endpoint. This was their primary endpoint of restoring hypoglycemic awareness. So it is becoming more and more durable, and the quality of life improves quite a bit by just having some function, despite being on a little bit of insulin.
Okay, thank you. So over to Bruce. Bruce, we have a question about A1C. Our A1C numbers, do these data still provide us with the most accurate indication of our management success?
Yes, absolutely they do, and no, they absolutely do not. It's a debate. So I think it makes a lot more, like from a clinical standpoint, as a clinician, I would much rather see continuous glucose data of some kind, a flash meter or continuous glucose monitor, which helps people with day-to-day management, but also helps me see patterns and see how well they're succeeding. The thing with an A1C, this is so hotly debated, but and I wish we could just use continuous glucose sensors, but it's not a perfect win either. So let's say I'm wearing a continuous glucose sensor for 3 months, but there might be a week or two where I'm just not wearing it. You know, I'm off my game. I don't want to think as much about my diabetes. My sugars are running much higher. I haven't worn it every single day. And usually if I were just not wearing it at random, you know, that day where like any other day, that'd be fine, but it's never at random. You know darn well those days you're not wearing a sensor are days you're a little less engaged in your diabetes management. So now if I go and I look at my estimated average sugars or time in range on my sensor, it won't be a real reflection of how much glucose exposure I've had. There it would be better to have a blood test, an A1C. So for example, in research trials, there's always this benefit to having A1C because we don't have to worry as much about what days people have worn them and not. But the A1C doesn't tell us how much time they spent at the extreme highs and extreme lows, and that's where a sensor helps us. So frankly, I think in clinical practice, I kind of love having both. And in research, I think often we just have to also use both and confirm the other. So I hope that makes sense. But just my official word is I'm much more excited about sensor data, but there's some realities where an A1C is more beneficial.
Yeah, and we know that not everybody has access to sensors, and that's something we'd like to see change. But so yeah, thank you, Bruce. Okay, we're going to go back to Cathy now. Cathy, can you talk a bit more about your process of identifying what components of the microbiome might be linked to type 1 Are you looking at what components are the most common in people with diabetes? And do you have an understanding about the mechanism of how these things might be linked?
Yeah, great, really great question. So to try to understand the microbiome, there are sort of multiple omics sort of studies that we try to do. The first thing that we do is we try to identify what bacterial species, even better, what bacterial strains that individuals have. And the best thing to do is to do that over time, because it could change over time if your diet changes or your lifestyle changes. And we need to do, traditionally we've done that using something called a 16S amplicon approach that will tell us kind of to the generous level, the genera level. So not very deep. It's not really telling us about individual species, definitely not about strains. So now that technology has really advanced much more, we like to do much more in-depth sequencing of the microbiota. That's going to tell us what's there, but it doesn't always tell us what it's doing. And so one thing that we're really trying to do now is to understand and to measure those small molecules that the microbiota is making, these metabolites. We can measure that in the feces. We are also looking to measure that in the blood or in the urine. And we're trying to, that will tell us a bit more functional information about the type of bacteria that you have. What we do know is we have quite a bit of information about different bacteria and what kind of metabolites they make. And we're starting to get a much better feel over what metabolites are very important in immune modulation and immune education. So what we're looking for now is really that, you know, the golden word is mechanism. And we're trying to understand what is the mechanisms that the microbiome uses, the cellular and the molecular mechanisms by which the microbiome and these metabolites shape and educate the immune system. We have quite a bit of data that looks interesting in animal models, but we need to translate that to human studies. And of course, it's much, much harder to look for mechanism in human studies. So we need to have more association studies. So that work this is work that's really been ongoing, and especially in the last 5 years, we're getting much better at associating or linking these metabolites, immune function. We need to have a really good look at the immune system development over time, and these the types of bacteria, the metabolites that they're making, and the effect on the immune system.
Okay, thanks, Cathy. For Andrew, so a question about islet transplantation. Why is this not available or recommended for people other than those who've not succeeded with other therapies? Wouldn't it be better to do earlier to prevent other complications rather than later when it's likely that these patients already have complications?
An excellent question. And again, it comes down to risk. So if a patient has relatively good control and their hemoglobin A1C is in range, and the therapeutic inputs and innovations that Bruce has described, then that patient will have reduced complications long term, the vascular complications, if they can get away with exogenous insulin in that target range. So that is really, that person wouldn't be a candidate for an islet transplant. So it still remains the ones that have that hypoglycemic unawareness and it's life-threatening and poor quality of life as well. So that is the main target demographic.
Can you just add a few words on the risks of the immunosuppression that's required when you have an islet transplant?
Sure. For the risk of immunosuppression, well, again, those drugs are diabegenic, so they're going to harm the graft, which will put you at risk of having another transplant. And when you have that, you're exposing these individuals to more HLA antigens. And unfortunately, a lot of patients with T1D require a subsequent, potentially a kidney transplant. So it would limit the donor pool of if you unfortunately needed a replacement kidney, as well as there's a chronic risk of infection and certain forms of cancer with lifelong immunosuppression. The drugs are getting better, but they are still very toxic to the body and the cells that we're transplanting.
Okay, thank you. I'm going to put this question to Bruce, but others can also answer. So how can you encourage people living with type 1 to join research as participants? And how do you think we can best reach the community to ensure we have a patient experience at the forefront of research? Super important question.
Yeah, you said you're going to answer it in part and then, or shall I begin?
Oh no, I'm also welcoming other panelists to answer, but I'm going to pass it to you first.
Okay. Yeah, how to get more engaged in research. Well, I mean, to be honest, the main thing is keep in contact with a care team, you know, your endocrinologist, your diabetes educators, and know what kind of research is going on in your area and in other areas. But ultimately, what we need is an amazing kind of likely online community where people can check in and they can even offer data about themselves so that researchers could see whether people could be eligible for their research studies. So basically a community of people with type 1 diabetes, as well as researchers like really dedicated to transforming type 1 diabetes, who are kind of together and able to communicate with each other. And that thing is on its way in Canada. And Sarah is heavily involved in this as well and helping with funding aspects of it. And it's called Connected, or will be called Connected. And it's an online community to connect people with diabetes and researchers. So stay tuned for that. But for now, do a lot of Google searching, check in with jdrf.ca, check in with your care team. And since we're in the last few minutes, I'll just very quickly, if you don't mind, Sarah, I'll just say now rather than at the very end, like thanks to Connected in Motion for putting this together, you know, to Kendra and to Shannon today helping out. But like, along with what I've just been talking about, I'd like to just say a huge thanks to Sarah. Like she's brought so much personality to JDRF in Canada since she's come on. Like it's just really transformed things and being involved in events like today. So that's my personal thank you that I want to sneak in before we run out of time.
Thank you, Bruce, for those kind words. And just looking at time, I do think we have to close the Q&A, I'm afraid, but we will look to see if we can answer some of these questions to individuals after the event. I'm also just going to share my screen here. I have the opportunity to mention a key fundraiser for JDRF that's coming up. And I would like to make a bid that everyone please support or continue to support type 1 diabetes research in Canada. And one way you can do this is by checking out our new virtual online event, jdrfparty.ca. And this money is all going towards research. So thank you very much to Connected in Motion for inviting us to join in this webinar today, to all of you for tuning in, and most importantly to our panelists, Cathy, Andrew, and Bruce. Thank you very much for enlightening us and sharing your research stories.
Thank you very much for inviting me. It was really a pleasure.
Yes, thank you, Sarah. And Connected Motion is fantastic. Learned a lot as well.
Me too.
Thanks,
Thank
everyone.
you. Nice to see you, Cathy.
Yes. Thank you all so much for being a part of this discussion. Just based on the comments in the chat and again on Facebook, the community is feeling really engaged. So I just wanted to thank all of you, Dr. Linklater, Dr. McCoy, Dr. Pepper, and Dr. Perkins for sharing your expertise, answering some really burning questions in the community. It's been so informative and engaging. And also thank you to our community members for providing such insightful questions and generating such an interesting conversation for all of us to follow along. So thank you for being a part of this Slipstream weekend with all of us.